webinar, A New Standard for 3D Cell Culture Heterogeneity. My name is Amarly Ferrigni, and I am a part of the team of Paralab Bio Spain. I am also speaking on behalf of my colleague Alexandre Borges from Paralab Bio Portugal. Today, we are going to have two keynote speakers, Dr. Andrea Cristaldi and Dr. Spartaco Santi. I am very happy to have you all here with us today. We know your time is very precious, especially in this time of the year, so thank you for joining. Over the next hour, we will cover the biophysical foundation and practical application of the new methodology developed to address heterogeneity in 3D cell cultures. Participants will be introduced to flow-based gravimetric technology, a label-free approach that integrates mass density, weight measurement with multi-orientation size analysis and spheroids and organoids. Today, the learning objectives will be understand the principles of flow-based gravimetric technology for analyzing 3D cell cultures, learn how to combine mass density, weight, and multi-orientation size measurement, enhance label-free characterization, recognize correlation with key biological features such as compactness, maturation, extracellular matrix development, and see how this approach can improve precision and quality control in 3D culture workflows. Get an overview of recent updates and practical applications in research contest. Before we start with these amazing speakers, I want to take some time to give you a brief introduction of who we are in Paralab Bio. Paralab was founded in 1992 by PhD student in chemical engineer with a strong scientific and technical background. Since then, the company has grown while maintaining its original vision and bringing advanced scientific technologies closer to the researchers. We have our headquarters is located in Porto, but we also have some office in Barcelona. In Porto, we have an in-house application with some certifications, like GMP certificated. Also, we soon are going to have an office in Madrid. Paralab, Paralab Bio is a Paralab Bio is a division of Paralab, and our focus is to provide technological solutions in bioscience research and industry. Our vision is to mediate the entire process of supplying and maintaining scientific instruments and equipment, but most important is to include providing technical consultancy, specialized training, and analytical service for material characterization. Finally, we are strongly committed to contributing to scientific knowledge dissemination, supporting our partners and the research community through workshop, webinar, and collaborative initiatives. This is part of Paralab portfolio. We have instrument and equipment and some service for multiplex analysis, cell development, cell line development, small animal imaging, structural analysis, interatomic, super resolution microscopy, intravital microscopy, bioprinting, flow cytometry, and live cells imaging and single cell tracking. Today, we are joined with Cell Dynamics to talk about spheroids and organoids. When we visit our laboratories or researches, the main challenge are variability between batches, difficulties in standardization, and need for reproducibility data. So the important, what we can extract from this webinar is how can routinely include a structural and biophysical information in your daily workflows without adding extra labels, extra toxicity or a huge burden in terms of time and analysis. This is where WA technology comes in and why we felt this webinar could be very valuable for you. Today, we have two amazing speakers, Dr. Andrea Cristaldi, okay, who is a scientific technology product manager at Cell Dynamics. Andrea joined Cell Dynamics in 2019 after more than a decade of multidisciplinary research in Italy and the UK. He is the co-inventor of WA technology and he now supports researchers worldwide with consultancy, experimental design, and co-author study. while driving ongoing research and development innovation. His background brings across disciplinary expertise to this field with a master in science in chemistry, biomolecular focus, and master in molecular modeling, drug design, and PhD in fluidic and bioengineering from the University of Southampton, UK. So he is perfectly positioned guide us to through the biophysical principles behind W8 and how they translate into practical workflows. The second one is Dr. Spartacus Santis, who is a senior researcher in the Istituto di Genetica Molecolare Luigi Luca Cavalli Sforza in Bologna. Dr. Santi is biologist with deep experience in advanced microscopy applied to neuroscience, immunotherapy, and personalized medicine. He has an extensive expertise in state-of-art imaging modalities, including TIRF microscopy, combined with resonance scanning confocal system, time-lapse imaging, light sheet microscopy, and super-resolution approach integrated with transmission electron microscopy. Beyond his research activity, he is deeply involved in scientific training. He teaches several courses in advanced optical microscopy and serves as coordinator of the Scuola di Microscopia. He also leads Digital Microscopy Center at the Rizzoli Orthopedics Institute, where he oversees cutting-edge imaging platform and promotes multidisciplinary research collaboration. Today, he will share with us his scientific journey with using WA technology and how integrating biophysical readouts has impacted the biological relevance and reproducibility in his 3D culture experiments. This session will last around 60 minutes. Please feel free to send your question anytime using the chat at the end of the meeting. We are answer them, and the session will be recorded and we will share at the end the main resources afterward. With that, I am delighted to hand over to our first speaker, Andrea. The floor is for you.
Thank you so much, Amarlik. Thank you, everyone. First, just give me a check if you can hear me. Everything gets okay with the audio? I guess so. I will take this as a yes. Thank you, Claudia. Thank you, Pedro. So I'm going to go to share my screen and give me a quick confirmation if everything is going all right about the sharing. How it is okay. Got a lot of information there. So thank you so much. Well, let me start about what I would like to share today. But before, I would like to thank the Parla Bio team and also Dr. Santi, who is a researcher that I really estimate. I love his work and the impact that he's having in the 3D cell culture field. So I'm very grateful to share the floor with him today. And my title of the conversation, as you can read here, it's about biophysical characterization of 3D cell culture. I want to give you a different perspective of if some of you is working on 3D cell culture or what you can do. So I've designed this presentation in order to be divided in multiple stages, I would say. The first part would be about the scientific gap and the rationale that we are covering. Also, I would like to talk about what is the W8 innovation, which I will be happy to be pronounced as weight. And then we will give you examples about the workflow and how this can be impactful from the beginning to the end. And then I will give you some examples of published results. Actually, I will share some unpublished ones as well in order to spark your curiosity and let you understand or have a new idea of how you can use this type of characterization for your work and to improve this. And obviously at the end of the time, after also Dr. Spartaco Santi presentation, we will have some time for the Q&A. Let's go first for the concept. As you can see from this title, we are actually about to shift the paradigm of size analysis, because anything that it's working on 3D, it is very much connected at the moment still with the very useful, obviously, analysis, they are based on bright fields. There are actually two sides of the conversation. The first one, which is taking care about the bright field analysis, when who's working with 3D cell culture is monitoring maybe size distribution, a little bit of the morphology and so on during the growth process. It's one aspect that's very simple, very easy to do. But if you think about this, the entire conversation now, it's about three-dimensional object. And when you're analyzing a bright field picture, I'm talking about very basic bright field analysis, other than getting the size and the diameter with software like ImageJ and things like that, you are still playing with a two-dimensional related picture. So in order to actually gather very detailed information, we need to move to the other side of the spectrum with something like confocal imaging, deep imaging in general, fluorophores analysis, dyes, and immunohistochemistry, those kind of analysis, which are drastically more detailed and important, obviously. But on the other side, it's also a little bit more difficult in terms of technology to be used, time consuming, resources, skills of the operator, and so on. What we noticed during the last probably 10 years, the cell dynamics has been created, and the last few years regarding the weight system, is that there were not technology in between. Something that keeps the same simplicity of a two-dimensional bright field with automatic analysis, but it gives you more 3D related results. Because again, we want to work with three-dimensional objects. And so we have to consider different perspectives. The question you might have is, how do we do that? That would be a very crucial question. And therefore, I want to share an example. Let's say you are producing some spheroids, for example, from 96 well plate, which is one of the most common basic 3D cell culture we can start. Whenever you are analyzing some size distribution, you might have some information like this, maybe two-dimensional derived diameter by picking ImageJ, length and things like that, from which you have a standard deviation analysis. That's absolutely fine, but if it is just a piece of information, what we thought is, why don't we use gravity to gather other information that are more related to the structure of the object? So we have designed a weight system which is capable of very simply picking your organoids, move them into a vertical flow channel, let them drop, and then track the velocity of falling to derive other information. So the first advantage is that we're now analyzing the size from two different optics, but the object, it's floating into a solution. So it's not, let's say, lying on a spacement like a Petri dish or something. And this makes the object in their actual natural morphology and sizes. So the first thing that we can do is averaging multiple picture to frame, and the average of the size distribution be very much more precise, I would say. But the key difference is what happens next. We leave the object falling down the vertical flow channel, and what we can derive is their mass density, which is directly related to compaction. Now, why this is important? Because you're passing from only having the information about the diameter to also exploding this information in another layer of precision, I would say, which is compaction. So now you can distinguish between objects that have similar sizes but different compaction. And as you can imagine, everything that is related to the biology might be absolutely correlable with this, because this is one of the biggest bias in 3D cell culture. They're much more reliable to be correlable to human translation, but they're also much more variable and hard to control in their heterogeneity. I can give you a very stupid example just for the sake of the concept. I can bring to you a bowling ball and a basketball that have the same diameter, But I don't think you will consider those objects as similar just because the diameter is the same. Because if you cut them, the cross section will be completely different, and therefore all the kinetics and permeation and so on. So let me go through planning a workflow. So it would be nice to understand if this is important for you or if this is already sparking curiosity for you. But in order to put this in an actual workflow and let you understand how you can play with this new value, I've decided to prepare for you to divide the workflow from the initial stage to the mid stage to the final stage so you can understand where to use it. Let's start from the initial stage. The first thing is that we have unlocked a distribution in multi-parametrical analysis, meaning that now you can correlate size with compaction. This means that you can design and you can understand a lot of statistical analysis that you can do for the simple fact that you have two variables that are correlated. That's the first thing. We're gonna go a little bit more in details later on. The second aspect, it's another big problem for researchers, meaning if you are preparing a batch with your 3D cell culture today, and the batch that you will do the next week, are they comparable? Are they the same? Do you have variability in a batch to batch differences? This is what we can do by using our quality control system, meaning that you can build a data bank of your structural data of your spheroids and organoids. So you can have size over mass density over weight, and then you can decide whether every batch you're producing is actually comparable. Therefore, you can make decision from the beginning of your workflow. For example, in this case, if you have a batch that doesn't overlap to the most important representative portion of your distribution, you know from the beginning that this is probably a batch that is worth not using, or that you have to do some variation in the protocol. And this is just the initial stage, meaning we are only analyzing one population of the object in an automatic manner, no labeling, nothing special in the preparation. When we go into the mid stage, things are getting a little bit more interesting because now you can do the same statistical analysis of one single population, but you can start making comparison. For example, you can monitor the trend between different time points of maturation or different seeding numbers, different patient-derived, different co-culture versus monoculture, anything that you believe might go into a different structural distribution can be monitored easily by analyzing about 20 spheroids, which is a good portion statistically representative of a normal 96 well plate population. And now in the mid stage, it's the moment in which you can understand that sometimes, very often actually, having mass density variation or size to mass density variation means something from the biological point of view. In the next slide, I will show you some examples about what it could be the correlation, but to give you a perspective, sometimes mass density is correlated to the cell number, but some other time it's correlated to the necrotic core, for example, if you have larger organoids, or to the cavity if you have some cystics or you have lumen inside your organoids. It's correlated to the extracellular matrix, for example, which is also very important. It's correlated to permeation from the cross-sectional gradients of compaction that you might create in a certain stage. And it's so interesting to see how a simple analysis can guide you into your workflow and being able actually to make better decision and also save time and money. Why not? By deciding what's the next step you want to do. Recently, we also coupled this with some multivariate statistical analysis. We've published just a few weeks ago, maybe last month, on Nature Scientific Report. It's a very interesting paper. We try to play with these values by using very common PCA, and it's very interesting how different structure will be positioned in a different part of a graph just by giving you the information of how the structure could be different and how this is important for any drug testing, for example, or any other type of activity you want to do. Now, imagine you have this information. You analyze your population in 1 hour, you get about 20 spheroids reading, you can understand their distribution, you can understand the quality, you can monitor the trend between different variation that you do, either could be number, treatments, you understand the compatibility or correlation with the biology. Now you need to go for confocal, which is, or you maybe need to go for deep imaging analysis, which is crucial aspects. And the key other function of this technology is that allows you to select and sample, meaning to separate only the representative samples that you want based on size or mass density, or a combination of the two, meaning you can potentially separate the organoids that has a same size but higher mass density from the one that has same size but lower mass density, and see in imaging how they correlate. This is precious for everyone that is working on imaging analysis because it's important to select a analytically representative sample for your population. Now, this is the entire workflow. Let me go quickly through how it can be used on each of these applications. Let's start from the structural variability. What you see here, it's an example of what I mean when I can tell you that having a second variable gives you a complete different distribution of the statistical analysis, meaning that each object that has the same size might have completely different variability in compaction. And this is obviously very important. Remember, we're talking about third dimension. That means that we have thickness to play with, and this is playing a role in a lot of kinetic information. You can have trends, you can have dispersion, you can have asymmetry distribution, you can have subclusters, meaning you can understand the subpopulation within your main batch. This is a typical view from organoids that have lumen, that in which the cavity is empty or is filled, for example. Or you have overall distribution. So a simple analysis gives you much more structural perspective of your organoids. We also have some quality control, as I mentioned before, meaning you can prepare multiple batches, you can select the threshold with the statistical standard deviation from your data, and you can create a data bank, a reference database, which means that every time you overlap a new batch, you can have a perception if this batch is good, is not good, what's the quality, it's deviating or it's not. And this will make you a guide for how to use in the next step. Maybe you have to tune the concentration of your drug testing, or maybe you can just decide to prepare a new batch because something went wrong. That's very important for saving time and resources. Let's say we're reaching on the next step, which is the mid stage. Now, Dr. Santi is going to talk about some examples, but I would like, for example, we'll talk about how we correlated some nuclear density with the mass density. I will leave the time to him, but I do also want to tell you a quick example in which the extracellular matrix plays a very interesting role. In 2023, this group published something. They wanted to compare some organoids from perinatal stem cell, actually spheroids, from monoculture or co-culture, monitor from day 4 to day 14. And they actually did the monitoring size and density. What you see is the size from day 4 to 14 in the monoculture. It's actually very similar, maybe a little bit smaller, but the shrinkage that you have for the co-culture is very strong. What they didn't know is that it was a plateau in the mass density from day 4 to day 14 in the monoculture, but it was increasing so much in the mass density for the co-culture. But the number of cells in this case was not really changing as much. So they started performing some fluorescence analysis on extracellular matrix components. You see the fibronectin here. But if you read the paper, they've done some collagen type 1, the laminin, the cadherin. And the point of the conversation is, if extracellular matrix is building up into my structure, then I have more compaction no matter the number of nuclei that I have. And this was very interesting to monitor with our technology because it was guiding and it was actually misleading the drug testing that other people can do. So having mass density to correlate size and number of cells with extracellular matrix is actually a very strong application. So strong that I want to give you another example, which is a little bit more basic. When you are running with thyroid spheroids, we've done this at the KISS Europe in Germany. They were preparing from 96 well plate. Very simple concept, same day of analysis, same maturation time, just two different seeding numbers, 10,000 cells versus 30,000 cells. They wanted to see difference in mass density, especially thinking that the 30,000 would have been more compact. Well, we noticed a very big differences in size. Size is about 370 for the 10K and about 500 for the 30K, but there was no difference in mass density. So we proposed about the ECM. We asked if it was possible that the 10K were anyway producing faster kinetics in the ACA information. We were on the imaging facilities and they brought us two pictures, which I am very happy to share here. Those are some scanning electron microscope pictures of the two lines of organoids. And as you can see, of spheroids, sorry, as you can see, you have the 30K here, in which each cell is separated and covered with a tiny layer, I would say. Whereas the 10K are completely embedded into the structure of the extracellular matrix. And this is one of the most interesting aspect of knowing mass density. But not only that, we also have correlation with permeation. You have to think that if an object it is more compact, it's more likely to have other infiltration cells to struggle into the permeation. And I will suggest you to have a look at this other paper that we have published in Frontiers in Immunology in 2020 about trying to track NK cells infiltration, which are labeled in green, in different spheroids type from colorectal cancer, in which we have differences in mass density. To be quick, obviously, I'm just giving you an overview here. The SW620 line, which was the highest in compaction, you can see that the NK cells, which are labeled in green here, they really struggle to permeate throughout the entire structure, whereas for the DLD1 spheroids, the permeation was more simple. So mechanobiology, it's strictly correlated to mass density, no matter the size. Other examples are about cavity. We're collaborating quickly this with Harvard in Boston, and these are pancreatic organoids, a very nice three-dimensional reconstruction, actually from Dr. Spartaco Santi. What we have done here, different protocol testing, separation by same size, but distinction by mass density. And this, after very deep, interesting 3D reconstruction, correlates with the volume of the cavity and the lumen about each of the organoids formation. And that's very important to see how researchers can be much more precise by knowing this. Although that wasn't published, what we have published in Nature Oncogene 2024, so last year, it's also a very interesting characterization with our technology on patient-derived prostate cancer organoids, in which we wanted to monitor the tumor cell invasion into the systems. What I want to do today is not go so much into the details, because I'm sure Dr. Spartaco Santi will do it next, but just to give you an impact of how this simple value can actually guide to make decisions into having a concept for three-dimensional structure information. And obviously, The key part, it's about separation. So whenever you have a certain distribution and you want to be precise on your confocal imaging at the end of your workflow, you can either separate by size or you can separate by mass density or for a combination of the two. This means that this portion of organoids or spheroids would be physically moved into another falcon tube, and you can actually go for precise analysis in which you will highlight the biology. Now, to conclude, these are some of the characteristics that you can find on our technology in terms of size limitation, throughput, and how to use it, but you can definitely find everything in our website. And I would like to conclude with some of the main users that are playing with our instrument and some QR code or information about the website that you can check. So for the moment, I think I took my time. I hope it was not so much confusing. I know it's a new value, and I wanted to give you an entire overview just that you can check on the published paper and the concept that we are sharing. And I'm pretty sure Dr. Spartaco Santi will go into details on some very interesting application. So thank you so much for listening, and I'm going to be here for any question you might have. Spartaco, I'm going to give you the floor.
Okay.
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 Thank
Can
you so
hear
much.
me, Andrea? It's
Yeah, I can.
Okay, I try to share my windows. Okay. Did you see my windows?
All good. I'm leaving.
Okay, good. Good afternoon to everybody, and thank you, Andrea, for your nice presentation, and Amarling for organization and invitation to this webinar. So in the next 20 minutes, I'd like to talk about recent application performed with weight. I like to call it weight physical cytometer, because I consider a particular cytometer, this device, for 3D cell culture system, and the correlation with biochemical data and above all imaging data, because I'm microscopy, so probably is my first job, the imaging analysis. So in the next minute, I showed 4 different applications in the field of oncology, regenerative medicine, metabolic disease, and the hand immunotherapy. For the oncology field of research, we use a model based on lobo cells. Lobo cells are human colon cancer cell line and are cultivated in ultra-low attachment plates. And for 10 days, this is the aspect of spheroids at low resolution inside the microcavity. The cell will cultivate for 10 days in the presence of crizotinib, different concentration, crizotinib 2050 nanomolar and 500 nanomolar. Crizotinib is an ATP competitive inhibitor of the receptor tyrosine kinase with a strong anti-proliferative activity. Was responsible for the activation of ALK, an aplastic lymphoma kinase, along the downstream of protein kinase cascade. And this is the imaging analysis of the different spheroids. So the control spheroids show a morphology very irregular, with many cell detached, and after the treatment, this phenotype reverse in a more compact, aspects, depending on the amount of crizotinib. So the effect of activation of ALK receptor on the colon cancer subtype 1 is published in Journal of Experimental Clinical Cancer Research in 2022 for the people who are interested to understand this mechanism. But for the application of weight, the first question, an important question, was understand if there is a different in the typical measurement of mass density, diameters, and weight if we fix the spheroid or take live spheroids. For this question, we collect two different samples, and here are represented the fluidic core chip, the core of the system. The direction of this channel goes from left to right, and the spheroids are here represented along the channel. So this system is able to measure in a range from spheroids in a range from 50 to 600 microns. And the data are here shown. So as expected, there is a decrease of diameters for both sample, live and fixed sample. The same for the weight is the same trend. There is a decrease for both, and instead the mass density display an opposite result with a significant increase in the density of the spheroid bulk in both situations. So the first conclusion is that the fixation protocol with paraformaldehyde did not alter the parameters under investigation, and this is relevant results above all for the usability of the device. But the second question, the second point is what does the mass density parameter represent for the spheroid phenotype? So to answer this question, we correlate weight parameters with biochemical quantification, in particular with protein quantification. Performed with the nano-orange assay, the protein quantification confirmed an overall decrease in protein levels in the same treated with crizotinib compared to untreated condition. So is in line with the diameter and weight. Probably is we can conclude that this is depending of the compactness of the spheroid itself. And so spheroid size and weight correlate with total protein content. Probably is due to the number of cells present in the spheroids treated with the crizotinib. But we perform also a image analysis. We start with a basic method, bright field analysis, and to calculate the optical density of spheroids. So the optical density is calculated as a ratio between the absorbance and the spheroid area, and the data are in line with the increase like mass density. In this case, optical density correlates with mass density, and mass density probably could be a measure of compactness of the spheroids. But we need another method to have more information in 3D. So we have to move in a better system, in a more sophisticated system. And for 3D information, we have three main methods to analyze the spheroids. The first is well-known is the multiphoton excitation that reduces scattering effect due to the light sample interaction because they use infrared laser light excitation. The second one is the light sheet microscopy. Illuminate the sample in a direction perpendicular to the direction of detection. Both methods are very sophisticated microscopy, very expensive, dedicated, and it's not easy to obtain for all the research lab. So we our method was this method. This method is combined the X-clarity tissue clearance system with the use of silicon oil immersion objective in confocal microscopy. We use this method for clearing the spheroids to remove lipids and homogenize the refractive index of the sample. That is the problem. In fact, the difference, the presence of lipids in a membrane reduce the light penetration, increase the scattering of the light. And the silicon oil immersion objective permit a very good resolution. In fact, we have a numerical aperture of more than 1. A very good working distance, more than 0.5 millimeter, and the reduction of the mismatch between the refractive index of immersion media and the mounting solution. The immersion media is 1.4 in this case, and the immersion solution is 1.46. And this mismatch is responsible for the aberration, spherical aberration typical of confocal microscopy. So these two methods is basically 2 steps. The first step is a polymerization. The hydrogel permits to create a link with the proteins, and then the clearing based on electrophoresis to extract lipids. So after that, the sample is very, very transparent to the light, and is possible to obtain a very nice optical section. After that, it's easy to calculate the number of nuclei for all the Z step. And the data are here represented. There is a decrease of number of nuclei after the treatment of crizotinib. And this is the image obtained with the segmentation to calculate the number of nuclei. So the conclusion is that diameter and weights correlate with the nuclei counting. Here I show you the analysis, immunostaining analysis with DAPI and phalloidine of control sample after crizotinib treatment. And in the bottom, the image, the signal from DAPI is represented in 3D rendering with ZDEP analysis, and the last object is represented in this nice rotation that show that all the sections are very clean and very nice with a good ratio noise, signal to noise. So the data of this study is published in PLOS ONE 2021, and the basic the demonstration of the basic of the functionality of weight in the micromachine 2020. Okay, now I'll move to the second application, the regenerative medicine. For regenerative medicine, one of the important challenge of this field of study is the cartilage repair. The treatment of cartilage lesion is a challenge, and a wide range of approaches are used to manage them. In the clinical practice, the procedure utilizes a non-pharmacological option, pharmacological option, or surgical approach. The surgical approach can only provide the pain relief and help manage the symptoms associated with the cartilage lesion, but they fail to repair the damaged tissue, leading to the formation also to a fibrocartilage tissue that can be deteriorated over time. So this limitation prompt the field to regenerative medicine, which try to restore and repair the cartilage using different approach, gene therapy or small molecule therapies like exosome, extracellular vesicle, and cellular approach. The cellular approach is very interesting because combine the cell therapy with the use of scaffold followed by growth factor treatment. And among cell therapy, a relative new approach for this for articular cartilage repair is based on the generation of spheroids. It could be used as the building blocks for tissue regeneration. So introducing this application, now the question is the use of adipose derived stromal cell as a model and the differentiation into chondrogenic lineage for obtain building blocks for tissue regeneration. So the two important questions we have in this field of interest is the first one: is there a correlation between the chondrogenic differentiation of spheroids and their cultivation method? And the second one, is there a correlation between the chondrogenic differentiation of spheroids and their physical parameters obtained with weight that can be defined a crucial point of maturation? So to answer to this question, this is workflow of our approach. First of all, the extraction of adipose stromal cells from adipose tissue, the expansion in vitro, and then the two methods of seeding with 3D co-seeded chips and ultra-low attachment plates. As you can see, the shape of the two systems are very different, the volume are very different. And then we analyze the spheroids after 7, 14, 21, and 28 days with three different approaches. The first one is measure the gene expression of SAX9, collagen 2, and aggrecan, marker of differentiation. SAX9 is the transcription factor of collagen type 2. A deep analysis in confocal microscope after clearing, and the weight physical cytometer analysis to calculate mass density, weight, and diameter. Looking the data of gene expression is well evident that after 21 and above all 28 days, the expression of collagen type 2 increased a lot for the cultivation in a chip, in co-seeded chip. And so the first conclusion is chip culture method improve the chondrogenic capacity of the cells. And this is also confirmed by immunostaining analysis in confocal microscope. The collagen type 2 in red is more expressed in chip cultivated spheroids, and here measure the fluorescence, average fluorescence of these two samples after 21 and in particular after 28 days. So this could be considered at the advanced stage of maturation, and is different if you cultivate the spheroids in a chip or in ULA plates. But for the parameters obtained from weight, very interesting that we in this statistical tool of minimum maximum normalization was used to compare biophysical characterization of weight, mass density, weight, and diameter for the different kind of cultivation, chip and ULA. and respected to the expression profile of the three marker of maturation. And it's evident the correlation between the chip at 21 days, the chip spheroids from 21 days, respect to the collagen type 2 expression. So mass density could be a most indicative biophysical parameter during chondrogenic differentiation and maturation, and could be used as a test to select the best spheroids that can be obtained during this cultivation. The data are published in Bioengineering 2023 recently. So after that, we can move in another application about metabolic disease. And for metabolic disease, we use adipocyte organoids, a very interesting model. In fact, for adipocyte organoids is a nice model, interesting model to study from the obesity and the correlate disease like diabetes type 2. And this is used to study for example, the efficiency of a different drug, and could be a test for novel therapeutic targets. The problem of the use of adipocytes organoids from this cell, that the cell have a big drop of lipids inside the cell. For this reason, the adipose-derived organoids have a floating behavior, and this buoyancy is a problem during the phase of analysis, also analysis in confocal microscopy. For this reason, we introduce the Folk extraction after the polymerization. This is a traditional method to isolate lipids from a biological sample using a mix of dichloromethane and methanol, and after that it's possible to extract the big drop of lipids. And the organoids, adipocyte organoids, now are ready for VO8 analysis, and after the clearing for deep imaging analysis. And if you compare these two samples from obese patients with or without type 2 diabetes, we can see a big difference in terms of weight and diameter, but above all in terms of mass density. And this is confirmed by deep analysis. In fact, there is a reduction of dimension in the presence of diabetes, and probably an increase of compactness, and is very evident in this large image. The nest where the big droplets of lipids were content before the Folk extraction. So the last approach is the immunotherapy. For the immunotherapy, we talk about CAR T cells. CAR T cells represent an advanced example in immunotherapy. In fact, the problem in a typical mechanism, a typical problem for the tumor is that the innate immune response of the host generally is bypassed and can inhibit the tumor growth and progression. For this reason, the CAR T cells are introduced in clinical trials to reactivate the immune response. The CAR T cells are T lymphocytes that are engineering to express this chimeric antigen receptor, CAR, which reprogram the specificity and the function of lymphocyte to recognize and destroy tumor cells. The procedure is complicated because after apheresis, the lymphocytes are isolated and engineered, introducing this receptor and then reintroduced to the patients. Currently, European Medicine Agency has approved 6 CAR-T-based products. 4 of them are against CD19 antigen, and 2 of them against the B-cell maturation antigen, BCMA. In my research, I use a CD19 target that is represented in this cartoon. This is the 2 players we use in vitro to reproduce what happens inside the patients. So we have CAR T cells, lymphocyte transformed and labeled with GFP, and target cells that is a K562 cell line, myeloma cell line, positive for CD19 and labeled with mCherry. So the red cell represents the target cell, the tumor cell, and the green one represents the CAR T cell able to destroy the tumor. After the attachment between the 2 cells, the CAR T cells is able to produce pro-inflammatory cytokines like interleukin-2, interferon gamma, TNF-alpha, and secrete granzyme and perforin to destroy the membrane of the host of the tumor and kill the cells. So in this experiment, we was very lucky because we identified just 2 cells, the green one CAR T, and the red one gave the target cell. And after a few hours of this incredible fighting, only one cell of CAR T is able to kill the target cells that make some bubbles in the surface, and at the end is killed and go away and detach from the plate. But normally what happened in this showing this time course of co-culture, the GFP move from the cytoplasm to the plasma membrane, and are generated this cluster of cells that represent the ability, the activity of CAR T cells to destroy its target. Oh, this okay. This is a very, very nice picture obtained with the holotomography microscopy before the contact and after the contact. Is well evident that the little cell is a CAR T cells and the target cell is the big one. And after a contact at the level of immunological synapses, the CAR T cell enter in the target cell. We study also, we perform also a bright field analysis to identify and to measure the formation of this cluster. This is related to the ability of the CAR T cells to be active against the target cells. So we can measure the area that increase if the cluster is big, and the track and the mobility. There's another parameter of activity during the immune response. This is just the graphical abstract of the last paper in submission, where other players are involved in this attack. In fact, are indicated many extracellular vesicle positive for CAR and positive for CAR and GFP, and for CAR and CD19. The function is not only a function, cytotoxic function, but probably is a function also for the activation of the other CAR T cells to go against the target cells. The cluster can be characterized with the weight, in fact, and the ability of the level of ability of this co-culture can be measured with the mass density. Mass density could be a parameter that identify the activation, the level of activation of CAR T cells. So in this case, represent and it's not easy. It's not easy to measure because it's not a really spheroids, but it's possible after 48 hours to obtain a cluster enough with enough density to be measured with 48 for weight. So for the conclusion, I show you that weight platform can be applied across a wide range of biological and biotechnological application, and represent a novel measure to quantify generally the structural organization of the 3D cellular system, and can be integrated in the workflow, your typical workflow, combined with biochemical data, with deep imaging analysis, and so on, to improve and enhance the quantitative outcomes and for a more understanding of the complexity of the 3D structure. With that, I'm finished, and I thank you very much for your attention.
Thank you very much for those excellent presentations. We are going to have now a Q&A session. We have here some questions. This technology is only for spheroid and organoids.
So let me check the, how can I go for the question? Is this technology only for spheroids? This technology is for spheroids and organoids within a range between, let's say, 100 to 600 microns. Yes, that's the actual main possibility to analyze objects. Absolutely. I don't know if this question from Claudia means that if she can apply on basically tissue engineering on things like that. But the ideal, it's spheroids and organoids in that range. Yes.
Yes. Finish answer. Okay, we can pass to the second question. That is, how long does a typical W8 analysis take to results?
Well, from the technical point, the weight system takes about 1 hour to analyze 20 spheroids in a good condition. And two results is basically instantaneous, because you have to think that you don't have any type of preparation. You just need to pick your organoids or spheroids and put them in a falcon tube. That you connect the falcon tube, the system automatically analyzes the objects, and you get the results at the end. So results are just for the time of the measurements. And you receive the results in a raw format of data. You also have some pictures of the analysis that you do, just in case you want to remove outlier. So 1 hour, it's roughly okay for about 15 to 20 spheroids.
Okay, thank you.
No worries.
Have you assessed evidence on viability impact post-analysis?
Yeah, we've done this. We have done this on actually 2 publications. The first one, it's about a work that we have done with the Alamar Blue viability test. What we have done, and that was also, I see Spartacus say yes, what we have done is we put some tumor spheroids in 3 different conditions. One condition was keeping them in the media. The other condition was maintaining them in the analysis solution, but not operating with the system. And the third condition was maintaining in the solution plus operating with the system. And then we did some tests. And if I remembered correctly, we had no statistically differences in the viability up to 72 hours. I will add something because that it's recent in the latest publication on the PCA, principal component analysis. We have done also some viability test on tumor spheroids, but with the Alamar Blue from, with the cell titer glow from Promega. And in that case, we got completely no difference in the viability for one type of tumor cell lines. The other type was going a little bit low, but that's also correlated with some disgregation for the structure. So, if you go too early in the analysis, Spartaco just mentioned something crucial. He said after a certain amount of time, you have enough compaction to be analyzed. So, for example, if you're running a drug testing and the drug has to roll to disgregate the object, obviously the object will be too much disgregated. I want to keep this absolutely clear. It's really depending from the cell type. If
Okay.
you're working with tumor cell types, there's no problem. But we did some analysis also on brain organoids, for example, and it's better to keep them fixed, or you can analyze them live, but it's not ideal to be replated. But in general, it's not ideal to be replated. The goal for selection is for the final endpoint, so that confocalist or facilities like Spartaco is doing, you can pick the one that you want to
Yeah.
correlate.
Just to add that sometimes the fixation increase limit the disgregation of the
Yeah, so the
object.
analysis is trustworthy.
And also to respond to the last question, this technology is only for spheroids. I showed that is also for a co-culture cluster of co-culture, but the problem sometimes is the level of aggregation, and
Mhm.
in this time we need to wait
Yeah, yeah.
48 hours to have a level of aggregation enough to the measure.
Actually, now that you're saying this, Spartaco, it's just popped in my mind that any object that can be within that range could be also used. We have done some experience on ovocyte, for example. Uh, so that's also something that can be done in one sense.
Thank you for your answers. This is a question that someone write me in a private chat. How suitable is W8 for core facilities or shared labs, considering it is easy to use and is compact size?
I will say maybe Spartaco is more capable on that part, but I will say it's pretty much because it's a very tiny instrument. You can put it even underneath the fume hood, and you can have quick access. So from every lab, like every groups that want to try to have understanding about structural variability is very good, but also for facility that have confocal imaging, that it's the endpoint key technology to use with the selection part could be very important. So I think it's ideal to be shared, actually.
Okay. Any comment, Dr. Spartaco? Your experience?
Uh, some years ago was a little bit difficult to use weight, but now is much easier, and also for basic operator. So the problem is the interpretation of the data. For that, there is nothing that study and try with different sample and understand what does it mean mass density and what does it mean, and insert your data with other data and combine them.
Absolutely. It's a new value, and the community is starting to know how to use it and the advantage of it.
Okay. Is this type of analysis also useful at the early workflow stage? Example, before a treatment or at the end before proceeding to downstream analysis?
Absolutely. That's exactly the slide that I showed. So from the beginning, when you can check the quality control of your batch, to understanding the distribution of your population, to understand heterogeneity, and then at the end to monitor treatment. So yes, from the beginning to the end.
Here, Maribel is asking to repeat the specification about the range of the size of the?
Sure, the size is between 100 and 600 micron. We can go slightly less and slightly larger, but that's the safe one.
Okay. Which is the maximum size of the spheroid that is possible to manage to keep all the good parameters? 600, right? Same.
Correct, yeah.
Romina said, unfortunately for cells from patient, the aggregation is fluctuating depending on the type of the cancer and metastasis. Ovarian is difficult to have a stable aggregation, as well as cupped cell line, for example.
Absolutely. That's one of the most important application. Maybe fixation can have a role in this, as Dr. Spartaco just mentioned. So what you could do, for example, um, variability is very big in patient, normal, but it's also important to understand patient by patient differences. So maybe you can try to pick the aggregate, try to have a fixation protocol that works for you, that keep them a little bit more, let's say, united in one sense. And so that can start to be analyzed. And so you can understand what's the size distribution, which is very wide, but you can also understand compaction distribution. And that makes you in a condition to be much more precise. For example, to decide the concentration of a drug for the personalized treatment or any other aspect. That's my point of view, but Spartaco is more expert on this aspect than me.
Probably a strategy with a polymerization could help and could help the maintain the aggregation, but it's not easy to introduce this part in the protocol without alter the mass density data. But anyway, can help to maintain the aggregation between the cells.
Sure.
Well, I don't see more questions. I don't know if you have any question. We can wait like 10 seconds. So if not, thank you so much for your
Thank you
talks.
to you.
Very spectacular. I don't know if that word exists in English, but thank you so much.
Thank you to you for inviting. Bye, Spartaco. I hope we meet soon. And thank you to all to the team.
Thank you, everybody, and thank you, Amari, for your organization. Very, very nice. Bye, bye to everybody.
Happy holidays to all.
Bye, everyone.
Bye. Thank you.